Okay, good afternoon. This is a special day for two reasons. One is because today, and they're interrelated actually. Today's we're celebrating the 30th anniversary of the Geriatric Research and Education Center in the VA, and many of you know the VA was a pioneer in promoting aging research, and the GREC program, which the national program was instituted in the 1970s, and then RFAs were opening for universities to apply, and several did, and Dr. Katz, who is a former chief of geriatrics, applied in 1987 and got our GREC, and he directed it until 2008. And Dr. Michelle Saunders directed it for a couple of years before I did. So we had a small celebration at the in the VA, and though the GREC continues to be a very important part of our research program, supports with salaries and infrastructure many of us and the Barshop Institute and an aging program in general. So, and it's a national program. There are 20, actually 19, because two just fused, including the one from our today's speaker. So we had this celebration of the GREC haven't matched with the visit of our speaker, Dr. Raymond Young. He has been a member of one of the top GRECs in the country, and when I became director, I was invited. I sort of invited myself to Michigan because I wanted to know. I wanted to see what a gold standard for a GREC should be. So I went and visited them. They were very kind in showing me around and helping me with our program. So Dr. Young has been part of our GREC for many years, and I'm very glad that he accepted to come. That is the second reason. It's a special day that he's our speaker. Dr. Young did his medical training in the UK, University of Liverpool, and then he also did some clinical training in Liverpool, and then a place I can't pronounce, so I'm going to skip it, in the UK. And then he came to the US to do his training in internal medicine in Detroit, and then fellowship in rheumatology and immunology at the University of Michigan. And he's also trained in geriatrics, so he's certified in internal medicine, geriatrics, and rheumatology. He's a Beeson scholar, and that's where I met, and has been a good friend and colleague of mine, and he's been rising through the ranks to be a leader in the field of aging, geriatrics, and immunology. He's been a professor for several years, the chief of geriatrics for 6 years, and the director of their Pepper Center and their geriatrics center for the last year or so. He's also held the Jeff Halter chair. Jeff Halter is a former director of geriatrics and their Pepper Center, and also very good to me, a good advisor. So I have a special connection with Michigan, and he's really done extremely well with his funding, continuously funded. Pepper Center heavily involved with their shock center and a number of training programs, over 100 publications. Dr. Young, it's an honor to have you.
Thank you. It really is my pleasure to be here and also get to meet people I know, and I have to say there seems to be a pretty well-worn path between Michigan and San Antonio. I'm seeing, a number of people kind of move junior faculty, really moving from Michigan down to San Antonio, and it's wonderful to see how successful they have been. And I have been telling people that the San Antonio Aging Program has always been an amazing program that is recognized nationally, internationally, and the work that has been done at this center has really transformed biogerontology and how we think about the biology of aging. And it's wonderful to have known Nick all these years, and the Beeson School is a little bit almost like a little club of people that you end up MDs and then now clinicians who early in your career where you get recognized and supported in a way, and in a way that was also kind of what brought me from the world of rheumatology, immunology into aging, and it has been really good to me, and I continue to be participating in a lot of the activities relating to the Beeson program, still very much part of their review panel as well as part of their advisory board, and that really has been wonderful. As Nick kind of alluded to, the way I would introduce myself may be a little bit different than the way that Nick may have introduced me, in that I'm someone who just couldn't stay in one field, and I love science, I love different things that I do, so I just wouldn't grow up, if you like. I always seem to feel like I'm a student, I want to learn new things, and so the medicine side about being boarded in internal medicine, rheumatology, geriatrics, and I'm also boarded in hospice and palliative medicine. That just wasn't a punishment. Doing one postdoc in training, fellowship training in one area just wasn't enough punishment. So I had to do more, and so on and so forth. So the talk that I want to talk about is that, I'm a clinician, and sometimes I tell people a clinician pretending to do research, and as we try to do what a lot of PhDs do, and sometimes it get really interesting, but fortunately I have a lot of good help from lots of people over the years, people who have just wonderful scientists that can help me do a lot of research. And a lot of the work, and I'll be giving some credit at the end as well. And I do both human and animal research. I love the translational band about research, kind of similar to what Nick has interest in. There are a bunch of us in the aging world who really want to connect the world of basic biogerontology to clinical side and benefit health. And it is an interesting way to do that, and sometimes it works well, and sometimes it doesn't work well. So what I want to do is to talk to you about one specific project that have been going on in our lab for a few years, and I just entitled it Obesity, Inflammation, and Aging. So let me see if I can get the... Okay, so as a clinician, I'd like to really start by putting my research into some clinical context. I always like to think about this as in what is the clinical context. So when we think about obesity and aging, one of the things that we can say is that we can look at our body weight and our BMI as one measure of obesity, and then if you do that, then you can argue that in humans, we peak at our BMI in our 50s. So when my wife kindly tells me I'm a 1.5 man she married, and not necessarily in a very nice way, she is right. I have gained weight ever since I was I was a medical student when I met her. And it wasn't because I was just have been eating McDonald's every day and I gained weight, but there's a natural aging process that goes on. But even though I can say, you know what, honey, this is my peak weight, and likely I won't gain any more weight, but the reality is that our fat mass and the body fat percentage continues to grow up, even though our BMI may be peaking at a certain age in my decade in here. That will continue to grow as I get older to my 60s and into my 70s. The difference is that even though my BMI may stay the same or start decreasing, there is where is the fat is an important difference, and part of that is that we develop more fat in certain areas, such as intra-abdominal, such as certain atopic fat depots, whether it be bone marrows, liver, or around the heart, and so on and so forth. And so this is the reality, is that we continue to develop, to have accumulation of fat. And we know that, at least in the clinical world, that obesity has been linked to many age-related illnesses. And in addition, it's not just about illnesses. We always talk about physical function, and that is the obesity certainly linked to decline in physical function, and also can be linked to frailty as well. So the so-called obesity paradox, and again, I'm giving some of the background because I know the audience is probably quite diverse. Some of you are more in the PhD world, and some are more in the MD's world, perhaps. The reality is that thinner is not always better. So I can tell my wife, it's not necessary that you want me to be the thinnest possible. And there are plenty of evidence that about the so-called obesity paradox, that particularly in older age, if you are slightly overweight or not necessarily really obese, you may actually live longer than people who are really thin. In particularly, it's almost as though the fact that the extra fat gives you that resiliency for you to, as you enter into some specific event in life. So going through an operation, having a heart attack, you may need something. So that is a little bit different from it in a laboratory condition, where our mice in our facility are protected for everything that you can think of, and they don't get exposed to things. But it may be that we need that extra bit of fat for us to be resilient to insult in our body. And the minimum mortality rate may be closer to the 25, BI around the 25 range, depending on an age. So this is a study that was published last year in Lancet, and looking at a global study, looking at various different places around the world, looking at the BMI correlation with mortality. And so in here, you can see the three age group, in the baseline age group, 35 to 49, 50 to 69, 70 to 89. And we're talking about the participant here is 1.2 million people. This is 2.2 million people. So this is a large number of people. And depending on which age, you see the classic kind of tick, or the shape curve, and in this age groups. And as we get older, that shape changes somewhat, so that it's more flattened, and rather than the sharp shape that you see. So when we are younger, it's probably true that when we are closer to the 20 or so BMI, we may be better off having the lower mortality. As we get older, it seems that we do need a little bit of increased body weight, and particularly fat that may not be all bad necessarily. So the research questions that I ask in this area are basically these three things. One is that I want to know whether a 20-year-old who decides to go and eat McDonald's for a year and get obese is the same as someone like myself who is still trying to, eat probably and whatnot, but still it takes so much harder for me to maintain my weight. Is that the same? How is diet-induced obesity different from age-related obesity and inflammation? Understanding that we think about the meta-inflammation world, that we know that a lot of things change in our immune system as we get older. So intuitively, you would think that there are differences between inflammation associated with diet-induced obesity compared to those who have age-related obesity. And the question has to be asked is whether this has related to the aging microenvironment versus the systemic factors that may be circulating around that could be play an important role in determining the changes that we see in age-related inflammation. So specifically, I want to say that obviously those people around here, particularly in Bioshock, all understand the role of senescent cells, which is a huge part of biology of aging. And I want to specifically say I would not be talking about senescent cells because I'm sure Jim Curtin probably come here every month or every other month to give a talk anyway. So there's no point of me trying to summarize his work for him when he can do it so much better than I can. So I wouldn't be talking particularly about senescent cells. And also the other things that I'm interested in is those of you in biology and aging obviously know about the so-called pillars of aging, the 7 pillars of aging that 2-3 years ago a paper was published and NIA got a bunch of experts, and it's somewhat controversial, but at the same time the idea is that are there specific areas of research that one should think about when we think about the biological basis of aging. And so there were 7 that were identified. And so my questions in here is the age-related obesity inflammation, how does it fit with these other processes that are thought to be at least important in the biology of aging? So those are the kind of 3 underpinning of things that I want to talk about. And before I do that, because I wasn't sure about the audience, I thought what I'll do is to talk a little bit about the difference between humans and animals. And many of you may have been to meetings where human researchers and animal researchers are kind of like this. They really fight and say, the human researchers said, the animal researchers, well, your work has no implication, nothing to do with humans, and the animal researchers say to the human researchers, clinical research, what you do is not even science, and you can just keep fighting. And we went through a lot of that. And so I think it is important for us to, those of us who are interested in translation to research, to think a little bit about that relationship about aging. What does it mean when we talk about mouse aging? So this is a graph that I thought gives you some useful thought to think about. So if we think that in animals, the average lifespan in mice, if we say is about 2 years. So depending on the strain, you can argue whether 2 years is right. And whereas in humans, if we say that the average lifespan in human, it's about 80 years old. Then if you just do a simple calculation, then you can say that, well, every 9 days I don't see my mice there in the facility, then the mice have essentially age 1 year of their lives. You can make that kind of calculation. But I think it's also important to think about, well, depending on what things you really are studying in animals, in mice, to really think about it. So we think about, say, during weaning. So for mice, the weaning period, those of us who do mice research know that it's about 3-4 weeks, and that's the weaning period. And for humans, it is about 6 months. That's when they, start eating solid and things like that. So if you want to say that calculation, if you're studying with the weaning period, then you can argue that, well, 1 year of human life will be roughly equal to almost 2 months of mouse life. So that's one way to look at it. Or if you're interested in looking at what about puberty when say in women, for example, or girls can now become pregnant potentially, as an example. And so we think about in mice, and we say, well, we bought from Jackson Lab, 6 weeks old or 7 weeks old mice, and that's what we get. And so you get a mouse that is about 42 days, and you can start breeding them at that point. And for humans, that will be roughly, we are talking about 11, 12 years old is when conception becomes possible. And if you use that as a calculation, then 1 year of human life is only equal to 3.65 days in that calculation. And then if you start thinking about what about adulthood, when we say, well, if we say, call it, say, about 20 years old is when we call someone an adult, and the same way then in terms of in mice, where we have the maturity is about 2 months or so, and many of you have probably have similar experience that I do. I will be a colleague will call me very excited and say, Ray, guess what? I have a cage of mice that have forgotten about it. It's some kind of transgenic mouse, and they're really old. What should we do for using those mice and the aging mice? And so, okay, well, how old are your mice? Well, boy, they're 12 months old. And I said, okay, so how old is equivalent of 12 months old mice in humans? Assuming, and then you say, well, it's not really that old when you think about this. So these are more like young adults and so on and so forth. So, a fully grown adult equivalent. So if you think about reproductive senescence, though, so in humans, we're talking about maybe 51 years or so, roughly, where we go through reproductive senescence in women particularly. And in terms of mice, depending on the strain as well, then you have about 15 months will be around that. And so if you calculate that, then 1 year will be equal to more like 8.82 days. So depending on what you're talking about, then the age group makes a difference. And so this is just something to think about. How often do we see our colleagues in, say, oncology and so on and so forth, and then they study a certain type of cancer, and then they use a mouse model, and then you ask, well, what age do you use? And is that an age-appropriate mouse model for your disease? And this is, I'm going to digress just one slide, and this is a study that we did a few years ago. And that was exactly the questions I asked. So that was the time when cell-based immunotherapy, cancer immunotherapy became very hot topic. And in a preclinical study, the results were spectacular. And yet when we went to clinical trials, then things just weren't going so well. And one of the things I asked in this study was, okay, you are using dendritic cells in dendritic cell immunotherapy to fight cancer. And in your animal study, you look it up, they all use the usual 6 weeks, 8 weeks, 10 weeks old animals. But we know that our immune function changes as we get older, and you're using that to fight cancer. Is it really that effective? So in this particular study, so if you look at these 3 lines in here, what we used was that we transfected a melanoma cell line called B16 with ovalbumin, and from there to make them more immunogenic. And then we basically take dendritic cells and then co-culture them with the antigen to make them react to the cancer cell, the melanoma cells. And then we implant the melanoma cells into the mice, and then we let them grow, and then we either treat them with young dendritic cells or old dendritic cells. So the black line here on top here represent the tumor growth rate. So they start taking, and then they just start growing like crazy. And if you look at the bottom line in here, the squares in here, and this is what you typically see in the animal study, that, boy, they work really, really well. You can see that the tumor size is pretty well controlled for a long time. But when we use old cells, or dendritic cells, old animals, then what we see is that it doesn't work nearly as well. So it's not necessary that, so what we're saying is that if you don't take into account age in some of your experiment, things are going to potentially lead you astray, particularly when you're relying on the immune system of the person to fight cancer. So that's just one of those papers that got me way more invitation to give talks and so on and reviews than the number of papers I published. And I said, no, this is wrong. Something is wrong. But this is something just for people to remember that it is important to use age-appropriate animal models as well. So when we get back to the story about the obesity, inflammation, aging, so one of the early studies that we did was to just look at the Adipose array and comparing young animals and old animals, and then young animals fed with high-fat diet, asking the question, are they different or similar? And you don't have to look at any of the specific genes. The point is that gave us a clue that even though we control so that the young high-fat diet animals grow to the size that is similar to the size of our old animals, that there are some differences in the adipokine profile. This is in the adipose tissue itself. And when we start looking into some of the morphology, one of the things that we see is that in fat tissue, and all the study that I'm presenting will be intra-abdominal fat. So understanding that there are human data looking at subcutaneous fat because it's easy to biopsy or look into muscle because you can get to it. Mice are different. It's a loose skin animal. So, and so looking at subcutaneous fat may not give you the really equivalent or what you might see in humans. In fact, in data that we have never published, we really don't see the changes that I'm going to present to you in subcutaneous fat as we see in intra-abdominal fat. So one of the things you see is that in fat tissue, there are these so-called fat-associated lymphoid clusters. So these are areas in the fat that a lot of immune cells seem to cluster around. It's almost like a lymphoid tissue. This is why it's sometimes called fat-associated lymphoid cluster. And when we look at it by morphology, in collaboration with Carrie Luman, who is one of obesity experts at Michigan, what we found is that we don't really necessarily see them more of this structure, but then they look different. They look a lot bigger. And this is just a graph showing the size that they have. And the other thing that you can do is to measure the vascularity inside the fat tissue. So one way to do it is to look at the distance to branching. So you can do a computer program that can do it for you just to measure what is the distance between they start branching. So by that measure, there is a shorter distance, so more vascularity in this animal, which is the same as what you will see in diet-induced obesity. And so this is one thing that is similar in terms of what you might see. We start dissecting out the cell. So the way that we look at adipose tissue is that it's not a homogeneous organ. So in biology of aging, I think we oftentimes look at the whole organ as though things are the same. In immunology, we tend to really drill things down further and further down into subsets. And so in adipose tissue, one of the ways you can separate it is that you can separate the adipocytes, which are the fat cells, and then when you take away those, there is the so-called stromal vascular fraction. And these are a mixture of cells, including some pre-adipocytes and other immune cells and so on. And then you have the adipose tissue macrophages. So those are the ones that had garnered a lot of research interest because they are the most abundant immune cells in the fat tissues. And so what we did in here was that we look at the adipose tissue macrophages content, and frankly, we don't really see a difference when you correct it for the size of or the fat mass. So we don't see a difference in the number of macrophages that are present in old adipose tissues. But what we do see is that their phenotypes are different. So just because they're macrophages doesn't mean that they're all the same. So for a long time, people have talked about resident so-called M2 macrophages and the pro-inflammatory M1 macrophages. And now we all know that it's more a continuum rather than truly a subset, and there are a lot more different ways that you can call them. And what we did find is that essentially the bottom line is that you do get a lot more macrophages that show the pro-inflammatory phenotype, even though the number overall is not any difference. And this is just some more data showing about the pro-inflammatory cytokines that are produced in the old adipose tissue macrophages, such as MCP1, IL-6, and so on and so forth. And then they also, interesting thing too is that this is the top graph in here is the adipose tissue macrophages, and this is the fraction that is the adipose tissue stromal cells, and this is the adipocyte. So about the time when we published this paper, there was a paper that beat us to it by a few months. But the interesting thing is that their data were very different from ours. And so one of the thing that we had to address that they didn't have to address is, well, why is it different from someone else who had presented the data? What they have found essentially was that there were a lot more of these pro-inflammatory cytokines that in aging that they felt came from the adipocytes fraction rather than the other fraction. So, but over, I think, the next subsequent couple of years, it became clear that the difference may have to do with the technique that they were using and likelihood, and every time, this is now a number of meetings, national meetings, people have pointed out is that likelihood, what happened was that the other lab, as they were fractionating the cells, some of these stromal-vascular cells may likely have died and actually have released the cytokines. So when they were measuring so that they thought were the adipose cytokine, was that it is actually relating to the other cytokines. So the magnitude, if you like, of the cytokines production is certainly higher in this fraction compared even to adipose tissue macrophages. So we think that likelihood that was an artifact in the technique that caused the difference. But we sure caused me some ulcer as we try to publish our data that that contradicts someone else's data. And the other thing we did was that we co-culture using the soluble fraction culture cells from the stromal-vascular fraction from the old cells and asked the question, do they also influence the macrophage inflammatory cytokine production? And the answer is yes. So something that is a soluble factor that is in the microenvironment in old age, in adipose tissue, that seems to be causing some pro-inflammatory cytokine production in the macrophages as well. And these are just some of the chemokine receptor data. Since the question is where do these macrophages come from, likely a lot of them originated from the bone marrow cells is what most people now believe. And they do exhibit a chemokine receptor profile that will be more inflammatory than homeostatic in that sense. PPAR gamma turns out to be a very important nuclear factor in adipose tissues. And at the time when we did the experiment and published the data, not as much was known, but it was known that PPAR gamma expression is important in the development of the regulatory M2 macrophages. And PPAR gamma sometimes is also called a glitazone. It basically responds to glitazones, and rosiglitazone is one of the diabetes drugs that have been used, but it also helped to restore PPAR gamma expression. So we find that in adipose tissue macrophages, in old animals, they have lower expression of PPAR gamma that correlate to our finding of less, if you like, M2 macrophages development. And then by treating in vivo the animals with rosiglitazone short term, long term, you can cause weight change, and that complicates things. So in short term, for a few days, we were able to show that that helps to restore the PPAR gamma level, and in return also helps restore the pro-inflammatory cytokines in these animals as well. And one of the things that we found that was kind of interesting, and again, I really wasn't sure what to make out of that initially. And when we look at the subset of cells, one of the things that stood out was that the regulatory T cells number was significantly higher in our old animals. And it is significant because by that time in the obesity literature, in the diagnosis obesity literature, everyone is reporting the opposite, that there are a lot fewer regulatory T cells in adipose tissues. But data was data. It wasn't something that we could just say, well, too bad, or we decided to just go ahead and publish it and hope that the reviewers didn't ask us to explain why we found that. And so we buried it in this draft and that no one could read. And it got through the peer review process and got it published. And then about a year later, there was a Nature paper that came back. So they found the same thing that we found, that in old animals, that you have a much higher level of regulatory T cells compared with young animals or even obese young animals. And so that was good that someone actually found the same thing that we did. And the other thing they did, they were way smarter than I am. And what they did was that they actually say, okay, let us deplete all the regulatory T cells in fat tissue specifically. So they used the fact that it turns out that PPAR gamma is an important part in Tregs in adipose tissue for whatever reasons, and they were able to deplete this regulatory T cell in these old animals, and show that in older animals, when you have depleted these regulatory T cells, that phenotypically they improved all the metabolic syndromes. So they were able to show that, and that's how they were able to get into nature, because they were able to do things that we weren't thinking about able to do. And within a year of our publication, that was really wonderful for them too. What we did ask was that it got me interested a little bit about the relationship between regulatory T cell and aging. And so this is a paper we published in Aging Cell. And at that time, and even now, there are a few studies suggesting there is a correlation between age and the number of regulatory T cells, that there may be more regulatory T cells in the peripheral blood in humans and maybe in mice. And that is the level of report that you see in the literature. So we want to ask the questions of, okay, well, why and are they functional? So in one of my earlier world, I used to do epigenetics work, and so I was interested in knowing that phospho3, which is a key regulator of regulatory T cell, both development and function, as to look at the phospho3 expression in aging. And so we did see a significant difference in aging. So naturally, it turns out that in T cells, we were actually, I believe we actually probably the first people in the world to report it that there's a epigenetic drift in aging. That was the day when there were 2D DNA gel type of things, and there's a PNAS paper that no one ever remember. It was a small number of people, but now it has been much more accepted. DNA methylation level correlating to health and mortality, and your biological age, and so on and so forth. What we find was that yes, there is the global DNA hypomethylation with age. But also that there is also changes in the methylation. That this is just a number of phospho3 cells that's increased, and that when we look at the phospho3 level, you can see that there is the change in old compared with young. And when we look at the phospho3, we did some power sequencing. Looking at specific nucleotides within the phospho3 promoter and enhancer, and what we found was that there was really no change in the promoter methylation, but all the changes appears to be in the enhancer region. So we kind of tie this a little bit with the epigenetics world when you think about as one of the pillars of biology of aging. We also found that, again, there was very little things beyond the correlated study number of cells and so on. And we were able to show that not only are there more of the regulatory T cells in aging, but that they are also functional in part, obviously, through the IL-10 pathways. And also in the T cell suppression assay, we wanted to show that by using both allogeneic and syngeneic bone marrow dendritic cell to show that functionally they are not just there because they are not working well and therefore you need more cells, but they are actually highly functional as well. And finally, there was a Nature paper suggesting that regulatory T cell function may be in part through its perturbation of the extracellular redox microenvironment. So we collaborated with the group that did this work and that published the work and at Michigan, and was able to show that indeed our H T cell, regulatory T cell from our H animals actually does the same thing, and part of it function maybe through perturbation again of the redox microenvironment when we measure cysteine ratio and so on. So the next chapter of what I want to talk about for a few minutes is to talk about the link between the other biology of aging processes. And one of the things that we got interested in was the relationship between aging, inflammation, and also the ER stress response. So we know that age-related protein misfolding disease have been linked to change in the ER stress response in aging, and that we also have data, and other people have published, showing that in diet-induced obesity, that the ER stress response can help regulate macrophage inflammation. So the questions we asked was whether aging affects adipose tissue ER stress response, and then in turn contribute to inflammation in aging. And this is just the kind of the typical pictures that we talk about with the three pathways, if you like, and then some of the key players, and like CHOP and ATF4 in here, just to remind those people who are not who don't do ER stress studies. And basically, a simple answer is that we were able to show that the ER stress response are upregulated in aging adipose tissue, looking at the various parameters. And then the not only that, but that the old cells are more sensitive. You do ER stress on these cells using chemical ways by something called capsaicin. You can see that they turns out to be they have an even greater response than you will see. It seems that in younger animals, younger cells, you're able to control that stress better. But as you get older, it's almost as though you are no longer able to have the fine controls, and so you have much more exaggerated response to stress, ER stress. And conversely, we can also inhibit the ER stress response, in this case using the chemical chaperone, PBA, and that we were able to then reduce that change by affecting ER stress. We were able to reduce the inflammatory cytokines productions as well to level that you really see in young animals as well. And the next thing that we start looking at is the macromolecular damage arm of the biology of aging, trying to see whether we can link the ER, change in the ER stress response to autophagy in the aging adipose tissue. So some of these have been done in some other tissues in aging. What we did was that we essentially just performed similar experiment, but looking at whether the similar phenomena also happens in adipose tissue in aging. This is the overall kind of overarching type hypothesis that we think how these may be linked. And again, those of you who don't do autophagy study, and I know there's some experts out there who know the field much better than I do, it's always very confusing because all the different names that are out there, the LC31, and then all the autophagy genes, 1, 2, 3, 4, 5, 6, 7, 8, and so on. And your backland, you have the Pi3C3, which is a key player in here, and then P6C2, which recognize a lot of the damaged misfold protein, and so the higher level you see represents increased impairment of autophagy as well. So part of this is just to show you the pathway, because basically what we did was that we start looking at the autophagy associated genes in the old adipose tissues, and what we found was that they do have a lower expression, significant lower expression in multiple of these genes, suggesting that the autophagy process is impaired in the aged animals. And so that is, we did similar thing when we induce ER stress response. We can show that that also can cause P62 accumulation in the old animals. And conversely, we use some pre-edible size cell line to do this particular experiment. We can show that similar things happens with ER stress linking with autophagy, and that if you block autophagy using another compound called bifidobacterium, then you can also elevate the ER stress response as well in tissue culture cells. And conversely, if you look at it in calorie-restricted mice, then we don't really see that through change in the autophagy genes they look much more like the young animals as well. So the last part, just for a couple of minutes, want to just talk about one of the new study that we have recently completed, and the paper is in press, and so it's actually online available, so I can feel I can now present the data. I heard that this might go into YouTube, and so, okay, I don't know. So I've talked to my lab people, okay, what are the things that you want us to present? Well, this is a paper that is available online now. And basically, we are asking another questions, and this is that, you know, in obesity, there's a lot, and in aging, there are a lot of lipid-derived products that are known to also activate inflammation. The question is, you know, does this play a role in age-related obesity as well? So there are these different lipid-derived products such as ceramides and something called diacyl glycerol, and there are also free fatty acids. So all this have been shown in many studies to be important in both insulin resistance as well as activation of inflammation, particularly through by interacting with the one of the natural ligand, Fat2A, and activate the TLR4 NF-kappa B pathway that can induce inflammation. And we also know that in published literature that TLR4 deficient mice have reduced adipose tissue macrophage inflammation and reduced hepatic steatosis, and that the Fat2A null mice are protected against obesity and insulin resistant with aging as well. And so in here, when we place our mice on high-fat diet, and by the way, we tend to be a lot more gentle in our high-fat diet, so we tend to use more 40%, what I call the American diet, rather than 50-60%, which is a lot higher, but get you a much more bigger hammer for your study. So in this case, we did see that just with high-fat diet alone, we are able to increase the TLR4 and Fat2, and as well as the inflammatory cytokine and MCP1 expressions as well. And disappointing to us, and this is one of those negative findings that we also found, but data is data, what can you say? You know, that we were expecting maybe we would see higher level of things like Fat2A level in aging, but we actually saw, if anything, the opposite is the case. And we didn't really see that much. We actually did see some NF-kappa B activation, but it was just disappointing that our hypothesis originally was that in aging you would see a lot more, but it just isn't the case. But nevertheless, we look at allowed the TLR4 deficient mice to age, we would see that they do have lower level of inflammation, whether it be the NF-kappa B expression and other cytokines level, IL-6, MCP, TNF, and so on and so forth. So they do appear to be protected somewhat from age-related inflammation. And similarly, when we look at the ER stress response, we do see that the TLR4 knockout mice do have a better ER stress and a better expression, if you like, of autophagy genes as well. And they also have, even though I said I'm not going to mention about senescence, we actually have some more data, but one thing I will just show is that these mice also have fewer senescent cells in the adipose tissue. And this is just a glucose tolerance test. I'm not showing any of the ITT. This show that there is some improvement in the GTT in the knockout old animals compared with the young animals as well. So this is the part that I just want to mention, and I didn't show any slide. I took away all the slides, but I wasn't sure. But these are things that we are working on and continue to. A lot of these, at least 2 out of 3 are now in manuscript form. And we start looking at the effect of adipose tissue-specific knockout from the PIPE3 C3 molecule, and we are looking at some of the macrophage subset and ER stress response, autophagy response. We also interested in some of the systemic factors. So yes, we are one of those groups that have now gone into the heterochronic parabiosis. So for those of you who are not familiar with that, basically we kind of connect the circulation between young and old animals, and then we are looking at the effect. The picture is kind of fun. It looks like a Frankenstein mouse when you have one smaller young mouse with one older mouse, and you connect them together and allow the circulation to flow. And bottom line is that there are factors that seems to flow from the young animals to old animals that can improve the phenotype of the older animals, but not so the other way around necessarily so much. So that there's some mild effect, but not as much in terms of the things that flow from old blood, old animals to the young animal that cause them to age faster. So some of you also know, for example, that there are now at least 2-3 companies in the country that are very brave, and now they are actually transfusing young blood and plasma, rather, to old humans. So you have, if you're rich enough, you can just join it and give these people $7,000 per infusion, and then you kind of keep getting it with the hope that you will remain younger. So this is very much like the vampiring story that you hear when you have an older person, then you suck the young blood, and then now you become young. So I don't know what it's going to be, but Tom Randall, who probably have been here too before, is at Stanford, is doing one of them, and I talked to him about it, and he said, well, they are very careful about the outcome, and then they're only doing for specific disease. I think they are getting ready to the data to be coming up soon as well. So we are looking at this from an adipose tissue inflammation perspective. And then obviously San Antonio is one of the ITP sites with the intervention treatment program. And Michigan is another site, and Jackson Lab is the third site. And so we do have, just like you guys, we have mice from these programs, and there are a number of drugs that have now been shown through the ITP to extend lifespan in somewhat of a gender-specific manner. And so we also are looking at some of these animals and looking at the effects on adipose tissue inflammations as well. So again, I want to just recognize people who've done a lot of the work, some of the dendritic cell work, particularly Annabelle Julius, as well as Sanjay Gad was key. Some of the macrophage work, particularly Emil Ghosh, in some of the work on ER stress and so on. He really plays a critical role in this. Teresa is a graduate student in the lab who has been a wonderful help in many ways, and I'm trying to get her finish her thesis in the next 6 months so that she can actually leave the lab and start her next step of her career. And Carol Lumen, I mentioned about his role particularly in some of the imaging studies, particularly Ruma Banerjee was helpful, particularly in some of the redox potential experiment that our lab just doesn't have the expertise. Funding agents, and thank you so much for your time. And somehow I saw in my schedules an hour and a half allocated. I never in my life I ever talked for 1.5 hours. So I think 1 hour is plenty of time. So thank you again for your attention.